Hello, everybody. Excuse me. Hey, welcome back to another great round of discussion. It is great to connect with you in this way, and I hope everything is going well. Hope you're having a good time, possibly over the weekend when you watch this. But who knows? You've got the option to watch it however, whenever it fits into your schedule. You just make sure you're taking care of it prior to us talking about it in the classroom. But again, thank you for opening this up. Thank you for continuing your educational journey. I greatly appreciate it. Love you guys. Appreciate you guys. And it's going to be another great topic. This will be chapter 4, as we've talked about in class, I'm sure, prior to ending the weekend. But this is chapter 4. This will be this video lecture series. This video lecture for this chapter will be in 2 parts, but that's more for the high school kids as opposed to the Crowder students. I should have said high school students. I shouldn't have said kids. But again, welcome, or thank you and welcome. Let's take care of that. Hey, welcome to the Nevada High School students. Thank you very much. Welcome to the Crowder students. Thank you for watching this. This is going to be a really good chapter. It's like every time I say that, it's because I just really love talking about this stuff. Really enjoy this stuff. It's really fascinating. And when you think about what we're going through right now with the coronid stuff, yeah, that's right. Coronavirus or COVID-19, however you want to say it, that brings up ideas of microbiology. And although the virus is a virus, so it's not a living thing, it does fit into the world of microbiology, which so do these little critters over here, the prokaryotic cells. But we'll get into all that in the appropriate time. But like I said, this will be a 2-parter. One, because the high school students, they won't need it all the first go around. So I'll break it up into 2 parts. That way I can separate theirs out and link theirs as needed. But for the Crowder students, just realize that you've got to watch this one and part 2 as well for your week. But hey, again, welcome. As you can see, I'm recording this on August 23rd. By the time the Crowder students watch this will be approximately, well, it certainly will be early to mid-September. And then for the high school students, we're talking this could be early October. Hopefully the school year is still intact and we're still getting to meet. But, who knows? Day to day, just crazy. But by the time we've actually gotten to this point, if we're in class all the time, we've kind of worked out some things. And so with that, let's get started. Hopefully I can keep this within 45 minutes. But again, as always, part of that time is you pausing it to write things down. And as you can probably see, there is a bunch for you to write down and to draw the cell structures, such as this right here and this right here. It won't be so important that you try to draw those and recreate those. We'll take the notes as in sections like this or like this right here. And then when it comes to the pictures like this one here, that's the individual organelles being pointed out, and we'll talk about each of those individually. So you'll have a chance to write down about those once we get there. So again, welcome, thank you. Hope everything is going well for you. Hopefully you are well, and it's life, health, mental, physical, everything. And again, appreciate you guys opening this up. And with that, let's get started. So we're going to start with, let's bring this down and go a little bit bigger. Okay, pull me up a little pen right here. Let's see. In the honor of, what do I got here? Let's go with this color right here, and that will kind of just lead us into some historical chatter. So if you think about the cell theory, now when we go through this, you're going to think to yourself, well, yeah, all that's pretty common knowledge, or pretty self-explanatory, or certainly easy to understand why we would think that. But you got to think back to the 1600s. Okay, you got to think back to the time when the church was kind of the prevailing, not only social, but cultural and historical center point. I mean, you had kind of two things going on back in those times. You either had walled cities where a king or some sort of monarch was in charge, and essentially all of life was wrapped around that scenario, whether it was the protection that the king provided, the leadership, or that kind of stuff, and even to the point where if you know enough about history and social studies, all the taxation and stuff that went on. So, and then you had the church as well, which was the ultimate authority in how things worked, and it simply went back to, well, that's the way God did it, and that's not right or wrong. I'm not here to open up that discussion. I'm just trying to paint a picture or remind you of kind of the mindset back then. So anything other than, well, that's the way God did it, wasn't looked upon, or wasn't looked upon favorably. And if you know enough about your history and science, both whether it was people who were deemed to be witches, or scientists that went against the God mindset, I mean, it just didn't go well. And right or wrong, that's just the way it was. That's the way it happened. So you just had to kind of think of it in that standpoint. So when people started being able to see how things actually work, that was big news. I mean, that was huge. If you can imagine a life where all you could see about the way the body worked was what you could see with your naked eye. Okay? The observations that were made, so everything, all the thoughts about the body were just through the filter of the naked eye. So they were trying to explain what we now know to be microscopic operations, with no knowledge of what a cell was, no knowledge of organelles or elements as we know them today. So it was big news when someone designed the first microscope and was able to see cells and atoms and things like that. I mean, that was huge. It was like having the entire world blind. Okay, this is kind of a metaphor. So you imagine the entire world is blind, and then one person is able to see and describing the colors and the images. And up until that time, everybody could hear. Okay, so they knew what bird songs were, but they didn't know what a bird looked like. That's what scientists were beginning to do for the world, beginning to open up our eyes to how things actually worked. And it wasn't that they were going, ultimately I should say, that it wasn't they were going against the church and God, is that for the first time we were able to see how they actually worked. And then if you believe that God made everything, well, he made those cells. So the scientists, even though the church might have thought, no, that you're going against what we believe how God did it. I mean, if God made the cells, then how they worked was also his formation. And they were just explaining that. So ultimately, it was just a misunderstanding, although detrimental at times to people. But anyway, that's beside the point. So, moving on. So I just wanted to paint a picture of when we talk about the cell theory, we're talking about it. We see it now thinking, yeah, easy peasy, I got this. That all makes sense. But when they introduced it, it was groundbreaking. I mean, it was front page news if there were newspapers. It was eye opening. However, whatever phrase you want to use there, to understand how the cells operate. So moving on, and so I chose brown because it closely resembled the idea of cork. Cork was the first main object looked at under a microscope to study, but we're going to talk about this stuff right here, the cell theory. And so you certainly want to write down all this stuff, all three of these, and put those to pretty good memory. Those are easy multiple choice questions there. So, but starting off with, all organisms are composed of cells. Okay, again, we say, well, yeah, we know that, but they didn't. Okay, they had no idea. This was kind of like trying to explain the airplane to somebody that we know today before there was any sort of locomotion besides a horse back in those days. Okay, all organisms are composed of cells. Cells are the smallest living things. Now we know there are things inside of a cell, but those aren't living. We consider cells the smallest living thing. And what was really huge back in those days, cells arrive only from pre-existing cells. And you may think, well, yeah, again, conception, reproduction, yeah, we got that. But they didn't back then. They didn't know that. It was believed that when there was new, like for example, conception, okay, it was just a gift from God was what their thought was. And that's not right or wrong, okay, either way. That was just all that they knew. Okay, that was them trying to explain a incredible process, amazing process, through what knowledge they had. And so for the most part, the church and the Bible, that was all the information some people had, and so that was the prevailing thought. But the idea, when scientists started doing experiments that showed that, yeah, when we put meat out and we cover it and it leaves out, it may rot, but we don't get all the flies landing on it, laying their eggs, and then new flies coming out of that. Okay, we don't get that. But if it's uncovered, then we do get that. And so that meant, or that just showcased that the flies had to lay the eggs, okay, and then that gave birth to new cells. The cells and the maggots just didn't arise out of the meat spontaneously. So that's the cell theory and a little bit of history, which I love talking about, the science history and the people and stuff like that. And if you ever get me in a microbiology class, which probably won't happen, but man, I spend way too many hours talking about it, but it's such, I just really enjoy it. Now, just for the review process, we're going to take a look at this diagram right here and just kind of remind ourselves about some stuff, because we can pick out some mitosis stuff in here, and for the high schoolers, we'll eventually get to those concepts. But for the most of these cells, if you think back to your EOC days, if you're either a high school student now or a recent high school graduate, most of these are in some form of prophase. That one obviously the very end of prophase. But most of the rest of these, you can just see some of the chromosomes, some of the organelles forming. But these two right here have very distinct mitosis stages. And if you really look closely, you can see these things forming in here. But what we're looking at, if we look right here at this one, okay, this is metaphase. Remember back, I'm just going to put meta here. People that I've had biology with, we always talk about, that's not metaphase. I went one too far. That's anaphase. Metaphase is when they're lined up across the middle. But anaphase, because they have been pulled apart to the edges, and this, you can somewhat see the ridges from the separating of the two cells, or the cell dividing into two. So that would be teleophase. So just a little reminder of mitosis, and also a little warning that eventually high school students, we will get to mitosis, meiosis, that kind of stuff. So that's anaphase, teleophase. So let's close this, and let's pull this over to right here. And now bring my pen back up. And now we're going to talk about cell limits. There are things that limit the size of a cell. And you can see here that, and you should just write down that the cell size is limited. And there are lots of variables to that. But we're going to talk about the main four right over here, and it's because of diffusion. Now, if you think about a cell, okay, it's got to get stuff into and out of the cell as needed. It's got to get rid of waste. It can also needs to ship, proteins or hormones or whatever it needs to send somewhere else. But for the most part, it's got to take care of itself. So it's got to get resources in. So if the cell gets too big, then diffusion can't happen. For one, the surface area is too wide, and it can't get the resources to the place it needs to quick enough because of the rate of diffusion. So that limits the size of the cells. If it gets too big, it can't get the resources across. Just if you think about if somebody, say on the edge of town, all the resources that a town would need, okay, could be found on the edge of the town. But the only way to deliver them is if somebody picks up the box and walks it to wherever it needs to go to. Okay. Now, if you have a little town that's 8, 9, 10 blocks wide, little square, then that person can get the materials, walk them over there, go back, get some more, walk them somewhere else. And although it may take all day, there's a chance that they can get all the supplies needed to each of the places it needs to get to. Now, if that town were 4 miles wide, and that person would not be able to, because there'd be too many houses, too many businesses, too many places for them to deliver the materials to. So that's the same idea with the cell. If it gets too big, the resources can't be delivered through diffusion. Okay, and that also plays into distance. If it's too wide across, again, we can't get the stuff there as a resource. Now the other two, temperature. Okay, if the temperature is higher, okay, now you want to think about kinetic movement, okay, cellular movement at higher temperatures. If we have an ice cube and we apply a little heat to it, even if it's just room temperature, it begins to melt and the atoms inside start to move faster. Same thing with diffusion. If the temperature is warmer, diffusion happens quicker. If it's slower, then if the temperature is lower, then diffusion is slower. It has that direct relationship. If you increase one, you increase the other. If you decrease one, you decrease the other. Direct relationship. Concentration gradient. Now this will be your first introduction to this word, but when we get to chapter 5, the next chapter, plasma membrane, you will hear about this a lot. Concentration gradient, meaning the amount of a particular element or material on either side of the cell wall, plasma membrane. If there is a bunch on one side and not on the other, then you have a concentration gradient. In other words, you have a bunch in one spot. In other words, there's a big difference in the amount of the materials. Now, when they have equal amounts, okay, you don't have a concentration gradient. Okay, but if you have a bunch on one side and a little on the other side, then you have a concentration gradient, meaning there's a big difference between the two amounts. So those are some things that affect the fusion, and which correlate right into why the size cells is limited. So again, be sure that all this you have in your notes. And I've been saying that. I should have started with a reminder of this. This is for the high school students. This is part of your grade. We got that. We've been checking on appropriate Mondays for a few weeks now, hopefully. And for the college students, that's just simply on you. The more you write, the more you'll know, the more you'll be equipped to work with us in class. But again, it's honor system for you guys. But for the high school students, it is part of your grade. All right. I also should have mentioned at the beginning what our topics were going to be. For chapter 4, we are going to, for the first part and the first video of this two-part series, we're going to lead up to talking about the organelles directly. So we're going to introduce cell theory, cell limits, prokaryotic cells, eukaryotic cells is in general terms. And then we will spend part 2 diving into the organelles themselves and some auxiliary information at the end. So with that, we will continue our march through part 1 of this video. Alright, so let's talk about all cells here in general. Okay, so get my pen back up here. Okay, so we are right here. And again, you're going to want to get this information, all this, and then for the drawing, you don't try to recreate it. You certainly can if you're an artist and you like drawing, feel free. But we'll talk about them enough in the notes and in class that this is just supporting what's over here on the left. So all cells, regardless of what type, have 4 common commonalities to them. 1, there's some sort of DNA containing device, whether it's a nucleus that the eukaryotic cells have or a nucleode that the prokaryotic cells have, but both of them serve the same purpose. The nucleus over here, the nucleoid over here, they contain the DNA, the chromosomes, and all that genetic material. So that's number 1. Number 2, there is some sort of cytoplasm. So that's this salmon or orange stuff in here, the eukaryotic, and it's more of a dull orange over here in the prokaryotic cells. Cytoplasm, as we'll talk about with the organelles, but it's essentially it's the space. Like if you're going to ship something, and you took a box, the cytoplasm would be all that space inside the box that the stuff doesn't take up. If you're going to send a pair of shoes, you bought a pair of shoes as a present, and you're going to ship them. You got that box, whether it's a Nike box or whatever, and you got the shoes inside, that paper around them, but all the space that those shoes don't take up, that's the cytoplasm. And it's a gel-like substance, and it's just designed for protection to kind of keep things situated in their spot and kind of cushions. So maybe you can think of packing peanuts or bubble wrap or that kind of stuff would be a slight metaphor for cytoplasm. Okay, the third thing that all cells have in common are ribosomes. Ribosomes are super, super, super important, and we'll talk about them in depth when it's their turn. But just in general, ribosomes are what make up, are what construct. Let me back up. Ribosomes are what read the RNA strands, which then forms the amino acid chains, which then is converted into a protein. So they are super, super, super important. And lastly, but certainly not least, because of its great importance, it's so important that not only do we talk about it here, we talk about it in the organelle in part 2 of this series, and it gets its own chapter in chapter 5, and that's the plasma membrane. And its job is super important because it decides what comes into and what exits the cell, and there is no greater decision made than that right there. And great lengths are taken, not only for protecting the cell from things on the outside, but a lot of processes go into place determining if something can come in and when something leaves. And we'll talk about all that in time. So that is the 4 basic common items or properties of all types of cells. So now we're going to close this, and we're going to slide over here, and we're just going to go slightly bigger. And I can't get any bigger than that, so what I should do is let's make this a little bit bigger so where it's easier to see. And then as I move down, I'll make things like maybe that one needs to be a little bigger too. But so all this stuff, all these words, I mean, you're just gonna have to write them down. It's just gonna be good, solid notes. So high school students, you're gonna have a couple of pages for us to check in class on Monday, or whenever we do check them. So we're gonna start with right here. So, of all the types of cells that are out there, or of all the cells that are out there, I should say, they all fall into 2 categories. They're either prokaryotic or they're eukaryotic. Okay, hopefully you remember from your biology days that we are eukaryotic cells. We have lots of cells. Prokaryotic cells are unicellular, meaning the entire organism is the one cell. Like, for example, bacteria, okay, one cell. Now, you may have a infection which has lots of bacteria cells, but each individual cell is the organism, one cell. So you may have a big group of them, okay? You may have a lot of them, but they're all individually one cell. Now, some things about prokaryotic cells that we want to know, okay. It does lack a nucleus and other organelles. This is a fancy way. Membrane enclosed compartments so they don't have anything like that. You know, no vacuole, no anything to store anything that contains anything, no compartments other than that nucleoid, which isn't a membrane enclosed compartment. It's just a place. It's like a closet that has their DNA in it. Okay. They also do not have distinct organelles. Okay. They don't have a mitochondria. They don't have, the lysosomes. They don't have all that stuff that we're going to have and we're going to talk about. That's just simply the outer, the inner, some locomotion stuff, as you can see, and then they're in their nucleoid, the DNA. Okay, whereas eukaryotic cells have a membrane enclosed nucleus and they have other compartments like a vacuole, and they do have organelles as well, and not just organelles, but lots of organelles. And you can see here some examples for prokaryotic cells. It's bacteria, and then for eukaryotic cells, animals, plants, and those two categories encompass almost every living thing. I mean, in the animal kingdom, that includes the insects, that includes, obviously the birds, and just about everything. You throw plants in there, okay, there's not too much left. Fungi would be the biggest part of that, and then all those microscopic things that live in like water and wherever else they may be found. So that's the two types. Okay, so again, you just got to write all that down. Okay, take care of that. All right, so the last two topics are introduction to prokaryotic cells, and then over there somewhere, eukaryotic cells. And it is the last two, but there's a lot of stuff still to cover. So we still got 20-25 minutes easy to talk about. So let me close this out. Let me move this up. Get my pen back. And so let's talk about prokaryotic cells. And again, you just got to write this down, you just got to write this down, just got to write this down. So just pause whatever you want to do and make sure you collect it all. But they are the simplest organisms. One cell, not much inside. They are low in structure, but they are incredible for what they do. They do it super efficient, super awesome mechanism they have to get done what they want to get done, and that's just simply to invade a cell and to use the cell's mechanisms to reproduce their cells to make more. So, and they don't do a lot, but what they do really well. All right, simplest organisms. Again, they don't have a nucleus, but they do have the nucleoid. They don't have a lot of internal structures. So in this venue, we talk a lot about what they don't have. But if you take a microbiology class, whether it's at Crowder or wherever you end up for college, you will spend weeks talking about what they can do. Okay, so but in here we're just like properties and structures, which they don't have much of. But microbiology, we talk about what they can do. They do have the ribosomes, which makes proteins that I mentioned earlier, and they got the cell wall outside of the plasma membrane. And in a microbiology class, you would spend weeks, multiple class sessions talking about that cell wall, because that cell wall is what makes them so difficult to deal with. So be sure you got that. And again, as I mentioned, let me make this one a little bit bigger, and we'll have to adjust it slightly as far as position. And so let me move this one over since I'm covering up that tail. All right. So now, bring my pen back up. We are here. So again, be sure if you're a high school student, be sure you catch all this. The cell wall. Okay. Bacteria cells possess a strong cell wall, and peptidoglycan. Man, it's always a struggle saying it. Yeah, I did it right the first time. Peptidoglycan. Now, or however you want to say it, or spell it, which for me is wrong every time. Now, the reason why bacteria cells are so difficult to deal with is because of that cell wall and the material that it's made of, because it is super reinforced, It is super strong. It is super durable. It is extremely hard to penetrate the cell wall because of that, and which makes them difficult to deal with. Now, but because they do have a cell wall, think about this, whatever education level that you are at, think about this. Because they do have a cell wall, that is what we attack when we use antibiotics or antibacterial stuff, to try and get rid of or defeat or remove or destroy bacteria infections. We use antibiotics because they attack only the cell wall, and that's important. Okay, that they attack the cell wall because our cells don't have a cell wall. One of the things that you will have to do in order to combat bacteria is to pick on something that they have that we don't. Because if we had a cell wall, we couldn't use antibiotics because they would destroy our cells too. So we picked on the cell wall, which is a very difficult challenge. Okay, very difficult challenge because it is so strong, and it gives them a fighting chance. And that's why when you have an infection and you get the prescription and the doctor, the nurse, or whoever you talk to tells you to take them all. Okay, don't just stop when you're feeling better, because you got to make sure you get every cell. Because as soon as you leave one, eventually you'll be right back to where you were. Because, one divided into 2, 2 into 4, 4 into 8, 8 into 16, and eventually you'll have the infection back. So you got to make sure you get them all. If you just leave one, that's one too many, because you're going to go right back to your infection in a couple of days. All right, the cell wall functions, certainly protection, and it does a great job at that. Helps to maintain their cell shape, okay, and it allows them or prevents them from taking up too much water, bogging them down. Okay, and then on some, the last thing in the bacterial cell walls, some of the prokaryotic cells have a capsule as well. So they'll have their plasma membrane, they'll have a cell wall, and they'll have a capsule, which gives them, and you can see it over here, right in here, the cell wall and a capsule, which gives them even more protection, as if they needed it already. All right, so that is the general structure. And again, this is the picture over here. But we'll do stuff in class to work with that, whether it's identification, fill in the blank, whatever it might be. But just to talk about that as in a review setting, we'll use the pictures to do that. So let me close down the prokaryotic or the pen tray and slide this up, and let's talk. And this is our last topic for prokaryotic cells, but man, it is a super important one. So we've got the flagellum to talk about prokaryotic cells, and then down below there you saw just briefly is a scale comparison of all cells and sizes, and then over there, as I mentioned, get my pen, over there somewhere is the eukaryotic cells, and you can see some of it kind of right there. So we got that picture, and then the eukaryotic stuff. Now, bacteria cells and any other prokaryotic cells got to be able to move around, and they do a great job. Matter of fact, you could almost argue that their mechanism for moving around may be their most sophisticated part, because once they get into, they're on your skin, and then you get a little cut on your finger. Man, they see an opening, they sense that opening, and they got to get inside, and this is how they move around. The flagellum, or flagella, is just one. All right. Okay, flagellum, and that's these. It's this long thing right here. It's almost like a little tail, except that tail can do amazing things. It is used for locomotion, but let's look at this basic picture here first. Okay, it's got this basal body, which we're going to talk about. All of this is what we're talking about over here, and you can see how sophisticated that is. But it simply spins, turns, rotates, whatever you want to use there. And so when that spins, that tail, that long tail, begins to whip. Okay, and you can almost think of it as a rudder on a boat, fishing boat, ski boat, whatever. That thing begins to spin and propels them in that direction. You know, it's behind them, just like with a boat. That if you don't steer it at all, whatever direction it's pointing in, that's the direction it's going to go, because it's just going to push it. That's just Newton's law, third law in action there. So it spins and creates that ability to move. Okay, now, we gave you a little bit of structure. We're going to come back to this big picture over here, but now we're going to go up here and talk about the different types or different structures of the flagellums that prokaryotic cells can have. Okay, just like in our picture over here. Okay, if it just has one, okay, that's a polar flagella. In other words, just one on one end. Okay, the word polar when it comes to chemistry, meaning there's more on this end than there is on that end. So polar means it just has one flagellum on one end. Okay, and just that alone would be a big help. But look at these other options. Lopetricus, a prokaryotic cell that has the Lopetricus flagella, now has multiple flagellas on one end, and that's huge because now it can steer. So think about that. Not only can it use a couple of them to get it moving down the road, okay, and if one gets you moving down the road, two not only would get you moving down the road, but would increase your speed of getting there. Three, okay, that's going to triple our speed if the speeds are consistent with each other. But if you have multiple, now you can move in a particular direction, but you can also steer. And you're thinking, well, how can they steer? All right, think about either being in a canoe or being on a floaty, a big lounge floaty in a pool, and how you can take either the oar in a canoe or your hand if you're on a floaty and just kind of paddle in the water and turn yourself. Okay, you can turn yourself in a complete circle just by moving your hand to the water, that we've all done, I'm sure, at some point in time, whether in a pool or in a canoe. You can turn yourself in a complete circle. So by using them in that manner, now you can steer. Okay, that is unbelievable that an organism this small, okay, this low amount of structure, but it's got this capabilities. So it can move. If it's got a Lopetricus, it can move in any direction that it wants to go. So if that wasn't cool enough or amazing enough, now think about an Amphitricus flagella. Now it's got one on each end. Okay, so it doesn't have the ability to steer like a Lopetricus flagella package, but now it can go in either direction. I hope you can see that. It can go in either direction. Okay, it can back up, it can go forward, go right or left, whatever you want to say it, call it. Okay, Amphitricus flagella. Now it's got options. I want to go over there, or I can go over there. Okay, that's amazing to have this level of structure overall, but this level of locomotion. Now, prokaryotic cells that have peritricus flagella, and you can see the image here. Okay, multiple flagella on each end. So now I can do it all. I can go either direction. I can steer in either direction. Okay, I'm the total package. I'm like the I don't know, LeBron James of prokaryotic cells. I couldn't come up with, and when I was your age, I'd have said, somebody would have said Chuck Norris or Michael Jordan or John Wayne perhaps, but this is a whole different era. So I guess maybe LeBron James would have been the name there to use. But anyway, super impressive locomotion for such a organism as it is. But now let's talk about, now we're going to be here, let's talk about how it works. First of all, we got to emphasize this down here, because we've talked in chapter 2 that the reason why it's so important to understand chemistry and how it operates within a cell. And here's a good example right here. This entire thing is powered or energized by chemical reactions. Okay, it doesn't get any more important than that. And you can see the plasma membrane here, and again, we're going to talk about that in chapter 5 immensely, but you can see the polar head. Okay, you can see the fatty acid tails in that membrane, and so that's your plasma membrane. And then we've got our cell wall here, okay, which is kind of the anchor part to all this. So you've got the part that supplies the energy, that's in the membrane. You've got and this is simply, you can just think of this as a simple machine. I mean, we've got our axle, we've got our wheel, okay, we've got our axle that spins. If you think about how a car works, you've got the two tires, which are the wheels, obviously, the axle in between, and then you've got the transmission that attaches to the axles. And when you push on the gas, okay, that spins the transmission, that spins that rotor, that turns the axle, and that moves the car. Okay, same thing here. So super important. Okay, super impressive locomotion for such a simple organism. I mean, it's amazing, simply amazing. And again, in a microbiology class, we would talk for weeks about this stuff right here. But that's all that we need to deal with right there. And again, the pictures we'll deal with in class as a way to review and talk about it and discuss it. So with that concludes our prokaryotic session. So let me close the pen tray so I can move this. Oh wait, no, I'm going this way. We got this picture down here too, this comparative picture down here to deal with. So this is just simply, and we'll put this up in class as well as talking about it. But it is pretty cool. If you think about the very smallest thing, atom, and if you're thinking, oh, I thought cells were, no, cells are the smallest living things. Atoms are, of course, obviously inside of cells. As a matter of fact, not only inside of cells, they're inside of things that are inside of cells. So you got your atoms and you got your lipids, making up the plasma membrane, which we saw up here. You got your proteins, okay, and then you got your various viruses, and you got your bacteria. Viruses are a little bit smaller than bacteria, you can see. Now, but to give you a relative comparison to eukaryotic cells, prokaryotic cells, this is the mitochondria. Okay, it's in a eukaryotic cell, and it's right there. Okay, and it's the same size as a bacteria cell. So that shows you that a bacteria cell inside of our cells, you can get hundreds in there. Okay, then you got your animal cell, plant cell, and we're about to talk about those in general. But hopefully you know that it's an animal. You can tell animal cell because it's oval or roundish, just almost like a chicken egg. And plant cells are more square or rectangle, okay, because of the cell wall. Okay, then the human egg, the female egg, a frog egg So there's your comparison there between sizes. And of course, chicken eggs, ostrich eggs, and you've got an adult female here. And all of these things you can see can be seen with the naked eye down to here. So if you had the ability, you could see a human egg. Certainly if you go to a pond and a frog has laid eggs, you can see those egg masses. And of course, we see chicken eggs all the time. And if you've ever seen an ostrich egg, those are pretty cool because they're pretty large. And of course, we can see people. Things that you need a microscope to see in general, or which would help, would be certainly the cells. Can't see those with your eyes. And you think, well, how can we see a human egg if we can't see a cell? Remember, a human egg is still millions and millions of cells. Okay, so don't forget about that part. And of course, the bacteria, the organelles. And if you want to see a actual virus, okay, this line right here, you've got to have an electron microscope, and of course, all those little components that are within a cell. You've got to have an electron microscope to see those. Now we will close out our tray and move over here to the eukaryotic cells. Let's back out just a little bit here. Adjust, because we're going to try to put it all on the screen at one time because it is a little bit bigger. And there we go. So we've got these things to talk about. So the only real writing part, okay, is going to be right here. Wait, I don't think there's anything down below here. Let's just double check, make sure. Yep, nothing else. Okay, so that's the only real writing part right there. And the rest of this stuff, as I mentioned, we will look at these in class pretty heavily. So don't feel like you need to write all this stuff down, because you don't. These are all the organelles, and we're going to handle those in part 2 of our lecture series on chapter 4. So we're just going to talk in general about these things. So in essence, we're probably 5-6 elements away from being done here. All right, eukaryotic cells. Okay, that's us. Multicellular organisms. Lots and lots and lots of cells. We're talking millions and millions of cells. Okay, so some key take-homes, and this stuff I think you may have already written it down. But be sure, possess a membrane-bound nucleus, and that's this organism right here, as well as this one right here. Okay, more complex than the prokaryotic cells, and that just visually you can see that because a prokaryotic cells will have a couple of these red dots that have this DNA in their nucleoid, and that's about it. Okay, and we got all this other stuff in there. We compartmentalize everything. Okay, each thing's got its own little section, own little place where it needs to be. The cytoskeleton for protection. So in the animal cell, that's all this little light green stuff. That's the cytoplasm. And over here, it's all this area in here where there's nothing at. That's the cytoplasm. And again, we'll talk about it in depth. But it's the packing peanuts. It's the bubble wrap if you were shipping something. All right. So again, the rest of this is the organelles, which we're not going to touch on now. The only thing we want to emphasize again is shape and structure. If you look at a cell, and we'll do this in class, we'll play the what type is this, animal or plant cell. You just want to get the idea that if it looks like it has a soft edge and it's kind of oval shaped, okay, that's an animal cell. If it's green, okay, from the photosynthesis process, and it's got a square or a rectangle look to it, it's a plant cell because of its cell wall. Okay, so keep those two things in mind. So, but again, we'll deal a lot with this in class, and all this information will be covered in part 2 of chapter 4 video lecture series. So with that, we'll start the goodbye process. Hey, thank you. Love you guys. Appreciate you guys. The mindset and the environment is a better word, that you are continuing your educational journey at this time is unprecedented. No one, no part of history have people had to go to school in this country in the environment that you guys are. So you are to be greatly applauded in how you're dealing with it and the things that you're doing to help in all these situations. I mean, you are to be greatly commended and treasured for what you are doing. And I appreciate the flexibility that you have given, which allows me to do what I need to do, especially, I mean, the high school setting, it's one thing, and actually since I'm filming, I'm doing this video prior to us even being together, I haven't experienced it with you guys yet, but with Crowder, we've had a week, and the ability to have my Crowder bubble is huge, and I can't say thank you enough for letting that happen and for being flexible with that. Thank you, thank you, thank you. It helps a lot. It means a lot. It makes it a much better situation. So be safe, be strong, be smart, enjoy yourself, do what needs to be done, when it needs to be done, how it needs to be done. And with that, we will say goodbye.